Let's start by talking about cellular respiration, the big picture. Cellular respiration isn't a process that you should think about in an abstract way because cellular respiration is how you get the energy to do everything that you're doing. As you sit there thinking, what is cellular respiration? You're using a ton of ATP. How are you using ATP? Well, you're firing nerve impulses, and those nerve impulses are only made possible by the fact that ATP was used to create certain imbalances of ions in the nerve cells of your brain. When you move a muscle, all of that's coming about through a molecule called ATP. And again, it's very easy to get overwhelmed by the details that you experience in an AP biology course and forget about how real this stuff is. And the reason why I love biology is because it illuminates what life is about. So let's go on and talk about cellular respiration. First of all, let's talk about its chemical equation. And that chemical equation is this. In cellular respiration, you take a fuel. And this is the stuff that we eat. So it's represented here as glucose. But it could just as well be starch that you break down into glucose, the starch that you might find in a potato or pasta or something like that, a piece of bread, some kind of starchy junk food. In your cells, you combine that with oxygen. The oxygen is the oxygen in the atmosphere, obviously. And then through a variety of processes that we'll talk about today, that gets converted into carbon dioxide and water. Those are the two waste products, and energy in the form of ATP. This is an exergonic process, which means that it increases entropy in the universe and it releases energy that can be harvested in order to do work. So in this diagram, what we have is glycolysis, which occurs in the cytoplasm. It's followed by a series of reactions that are called the link reaction. The link reaction brings the products of glycolysis into the mitochondria. The mitochondria is that remarkable organelle that is essentially a cell that's living inside every single one of our cells. It's not shown here, but mitochondria have their own DNA, they have their own ribosomes. They were once independent living cells that took up residence within a single-celled ancestor of the cells that we have now. And that gave rise to the eukaryotes. It's an extraordinary thing that really changed the universe because consciousness is really only possible because of the amount of energy that this organelle makes possible. So there's glycolysis, the link reaction follows. The link reaction is followed by the Krebs cycle, which happens in the mitochondrial matrix. And the mitochondrial matrix is sort of the cytoplasm of the mitochondria. It's where the DNA are, and it's also where a variety of enzymes are that take the products of glycolysis and ultimately break them down into ATP for an energy carrier called NADH, and also releases a lot of carbon dioxide. And then finally, the last phase of cellular respiration is called oxidative phosphorylation, and that happens along the mitochondrial inner membrane and the intermembrane space. So at this point, we basically have the geography of cellular respiration. We've got the extracellular space over here, we've got oxygen, we've got the mitochondrion with its inner membrane, outer membrane, intermembrane space, and the matrix over here. And that is sort of the where of cellular respiration. Now, let's elaborate on what I just talked about, and we'll talk about the various phases of cellular respiration. So we begin with glycolysis, and glycolysis means the splitting of sugar, and you see that happening in this side of the diagram, where a glucose molecule is broken down by a variety of enzymes. I have an entire tutorial about enzymes. We're not going to talk about them in detail here, but everything that happens is happening through the action of an enzyme. And that renders glucose into a molecule called pyruvic acid. Along the way, the energy in the glucose becomes NADH, which is an electron carrier, and ATP, which can be used immediately for work. That NADH is going to be shuttled eventually to what's called the electron transport chain, which is where most of the ATP in cellular respiration is derived. So that's glycolysis. Following that is the link reaction, and the link reaction is number 2 over here, and it brings pyruvic acid into the mitochondria. And this arc over here in orange represents the mitochondrial membrane, and it converts pyruvic acid, or pyruvate, into acetyl-CoA. Acetyl-CoA is yet another highly energetic molecule that's going to feed its energy into the Krebs cycle, but we'll talk about that in a moment. But along the way, that conversion of pyruvate to acetyl-CoA generates NADH, again, an electron carrier that will be shuttled into the electron transport chain. 1/3 of the carbon dioxide that you exhale is generated over here in the link reaction. So of every 3 molecules of carbon dioxide that you exhale, 1 of them is generated here. The link reaction is followed by the Krebs cycle, where acetyl-CoA is fed into a series of enzymatic reactions. And let's be clear, they're not actually doing a little circular dance. These are just enzyme followed by enzyme followed by enzyme. And the end point, you'll see, is the same as the beginning point, which is why this is a cyclical reaction represented in a circle like this. And the energy in acetyl-CoA, this 2-carbon piece over here, that is completely broken down. It's completely oxidized to generate NADH, an electron carrier, FADH2, and another electron carrier that we're meeting for the first time, and ATP. And that ATP can be used immediately for the cell to do some work. In glycolysis, in the link reaction, and in the Krebs cycle, we've generated all these electron carriers, mobile electron carriers. What does that mean? You can think of these as batteries that can be in a charged or an uncharged state. When they're in this form, NADH, FADH2, they're charged. But they have alternative forms that aren't shown in this diagram. NADH can also just be NAD. FADH2 can be FAD. And those are the uncharged forms. When you charge up a battery, you stick it in some device and you plug it into the wall and you give it electricity. Well, basically, if you give these things electricity, they wind up in their charged up forms. So they come over to the electron transport chain and they give up their electricity. They generate a flow of electrons, and through an unbelievably cool, mind-blowing process, that energy is used to generate ATP. And that's the end phase over here. That's the electron transport chain, which oxidizes NADH. When these things go from their low energy form to their high energy form, well, something's being oxidized to give them those electrons. These are now reduced. Things that are reduced in general have a lot of energy. Things that are oxidized have low energy. Rust is oxidized. Carbon dioxide is oxidized. And these have a lot of energy. They give up their electrons. They get oxidized. And that creates a flow of electrons that powers the phosphorylation of ADP into ATP. It's an unbelievable process, and we're going to break it down step by step in the remainder of this live review. I haven't seen any comments, though, in the chat, and there's a couple of reasons that are possible for that. But if you're just watching this and you're feeling a little shy, don't feel that way. Go ahead, put a comment in the chat. It'll make me happy. It'll make me feel as if I'm actually doing some teaching, which is something that I love to do and want to do in this context now. So go ahead, put a comment in the chat. Just say hello, tell me how I'm doing. Say, keep going, Mr. W. Thanks so much. I'm from whatever city you're from, and I would really love that. And looking there, just seeing if there's anything in the comment stream. And let's keep going. Okay. So that was the big picture of cellular respiration. Now we're going to get into the details. We're going to talk about glycolysis, the link reaction, and the Krebs cycle. And here's something that's a little weird. I've got a green screen behind me, and this cup is green, so it's showing up in a really bizarre color. Okay, so let's talk about glycolysis in some more detail. Glycolysis has three phases. There's the investment phase, there's a cleavage phase, and there's an energy harvest phase. Investment, when you invest in a company, you put money in. When you harvest your field, you get to pull out the riches of what you've planted. You get to harvest some energy. And to cleave is like a cleaver is like a kind of knife for cutting something. So those are the three phases: investment, cleavage, and energy harvest. And so during the investment phase, what happens is a variety of enzymes, and the enzymes are represented by these arrows here and not by these arrows here. And not every enzymatic reaction here is shown. There are, I believe, 10 reactions. I don't know if I have 10 arrows here. I might. But for AP Bio, you don't need to know the details at that level. All you need to know is that enzymes are controlling a series of reactions. And you need to know the three big phases: investment, cleavage, and energy harvest. So during the energy investment phase, enzymes take a phosphate from ATP, which they render into ADP, and they put that phosphate onto an intermediate compound. So glucose becomes glucose 6-phosphate. Not important for you to remember, but you can see that there's a phosphate over here that wasn't over here. Another enzyme, or two, takes a second ATP, breaks off a phosphate, and plants it onto this molecule to create this, fructose 1,6-bisphosphate. Fructose is an isomer of glucose. It's also C6H12O6, but now it's doubly phosphorylated. When things are phosphorylated, they gain energy. ATP has more energy than ADP. So this molecule is brimming with energy, and that enables the next enzyme to take it and cleave it into two pieces. That's the cleavage phase. You break this molecule apart. It had 6 carbons, glucose, C6H12O6, and now we have 2 3-carbon molecules. Truth be told, there's another molecule that shows up for a moment, but there's an enzyme that immediately converts it into a second G3P. So here we have this molecule, G3P, also known as glyceraldehyde 3-phosphate. It also shows up in photosynthesis. It's one of these important molecules that shows up again and again in biochemistry. During the harvest phase, a couple of things happen. G3P, loaded up with energy and possessing the initial energy that glucose had, is now oxidized. What is oxidation? Oxidation is loss of electrons. And the oxidation of G3P powers the reduction of NAD plus to NADH. And that's going to happen twice for each G3P. So it happens once over here, NAD plus going to NADH, NAD plus going to NADH. And enzymes, represented by these arrows, phosphorylate 2 ATPs. Now, by design, I'm not showing all the details here. So you just have to know that there are phosphates that are available in the cytoplasm, and they're used to take ADP and create ATP. So our net yield we'll talk about in a minute, but let's just talk about what our gross yield is. Our gross yield is, that's the yield before expenses. So we have made 2 NADHs, and we made a total of 4 ATPs. That's our gross yield. So what's our net yield? Net yield is your gross after expenses. So we net 2 ATPs, and we net 2 NADHs. Why 2 ATPs? As opposed to 4, because it cost us 2 ATPs in this phase of glycolysis, the investment phase. We invested 2, we got 4, our net was 2. That's glycolysis. That's all that you have to know for AP biology. Know that, you'll be able to answer any question, and that's probably true on any chapter test related to this material. If you're at the college level, it really depends on your professor. You might have someone who's requiring you to memorize all 10 phases, but that's really not what I'm here for right now. Okay, and we're leaving this with 2 pyruvates. These pyruvates, if you added their chemical energy together, they would have less energy than the initial glucose that went in, but they still have a considerable amount of energy. Glucose got a little bit oxidized in order to create this NADH over here, and in order to power the phosphorylation of these ATPs, but there's a lot of energy left, and therefore cellular respiration is going to continue. By the way, on YouTube, I've got a great song about this, my glycolysis song. I'll play it for a second. Anyhow, I hope that you could hear that. Would be great to get a comment or two letting me know if you could hear the music that's coming through. But I want you to know also that a fantastic way to learn this material would be to listen to the glycolysis song. There's a page with the lyrics up on learn-biology.com. And once you know the lyrics pretty well, then you could try this. So the lyrics are going to be all you, like this. Glycolysis is a series of reactions, energy takes glucose, a molecule of sugar, breaks it up into ADH and ATP. I should stop while I am ahead. Anyhow, I, because of the kind of person who I am, can't imagine something that would be more fun than to have a glycolysis party. Not really just a glycolysis party, a cellular respiration party. So if you're there out in YouTube land thinking that would be fun, just put a comment and let me know. Say, Mr. W, that would be so fun. I would love that. I'd love to hear that. And anyhow, again, don't be shy. Would love to hear some comments from folks in the viewing audience. So now, after glycolysis, there's the Krebs cycle, but in between, there's a reaction that links the 2 together. And that doesn't have a name like glycolysis or the Krebs cycle. Its name is the link reaction. And the link reaction, excuse me, takes this molecule over here, which is pyruvic acid, 3-carbon molecule, and converts it into a 2-carbon molecule. This is going to get oxidized. Its energy is going to be taken away through enzymes that oxidate it, and that oxidation is going to be used to power the reduction of NAD plus into NADH. So let's look at the details of that. This is the link reaction. And what happens, again, is that a pyruvic acid from glycolysis, over here at letter A, is transported across the inner and outer mitochondrial membranes into the mitochondrial matrix. That's this space over here. The first thing that happens is that enzymes snip off CO2. This is what's snipped off, and you can see CO2. If you know your functional groups, you'd recognize that as a carboxyl group, and that carboxyl group goes into the atmosphere, and you are exhaling it. So this is 1/3 of the CO2 that you exhale. Other enzymes, excuse me. Other enzymes are going to take this molecule over here and use it to reduce NAD plus into NADH. Spent battery, charged battery. That's how to think about that. And then finally, another enzyme is going to attach the 2-carbon fragment that's left. We took off 1 carbon over here, got 2 carbons right here, and it's going to attach it to a carrier molecule called acetyl-CoA, coenzyme A. It's a much bigger thing than is really shown over here, but you really don't need to know anything about it. You just need to know that it carries these 2 carbons onto the next phase of cellular respiration, which is the Krebs cycle. This 2-carbon fragment attached to this over here, that's a sulfur atom, that's acetyl-CoA. That's a molecule worth knowing because it's the input of the Krebs cycle and it's the output of the link reaction. So we've now talked about glycolysis and we've talked about the link reaction. So now let's move forward into the next phase, which is the Krebs cycle. Joseph, what are the enzymes involved in this process? Joseph, first of all, thank you very much. You know, I have to say, Joseph, I'm not a biochemistry teacher. I'm an AP biology teacher. And you've got it. Pyruvate, it's dehydrogenase, but that is excellent, and that's exactly right. So what you have is dehydrogenase enzymes, and I'm probably going to get a little bit out of my depth, but what they're responsible for doing is taking hydrogens from pyruvic acid, I hope I'm getting this right, and putting them onto NADH. So that sounds right. Joseph, it sounds like you know a lot of biochemistry. So if that sounds good to you, then let me know. And again, Joseph, thank you for your comment. If there are other folks out there besides Joseph, please feel free to ask me any question because that is what I am here for. So we're at the end of the link reaction. We're good to go with the Krebs cycle. So let's do it. So we left off at the end of the link reaction with acetyl-CoA. Acetyl-CoA. Wow, Joseph, you're making my day. Joseph from Brazil did a biology course. Your music videos helped me a lot in biochemistry. That's fantastic. Joseph, I am really, really glad to hear this. And the idea that I am teaching someone who's in Brazil is just filling me with joy and happiness. I can't tell you how happy I am about that. So let's go. And Joseph, again, I don't know if you're one of hundreds or if this is a one-on-one tutoring session, but if you have any questions, stop me at any time. Obviously, I'm reading the chat and I'll ask you anything. And frankly, if you had another question that wasn't even about the Krebs cycle, you know, I could jump anywhere on my website and probably put together a little lesson about that. So Joseph, thanks. It's really great to have you here. So Krebs cycle. We're starting over here. Each of these things that are drawn to look like enzymes are in fact enzymes. In certain courses, college courses, you might have to know the names of all of those enzymes, but you absolutely do not need to know that for AP biology. So here's an enzyme whose function is to take the 2-carbon fragment, acetyl-CoA, that comes out of the link reaction. And I want to emphasize, this little piece over here, that was once a part of glucose. It's still got a significant amount of chemical energy. It's a reduced molecule that can be oxidized, and that oxidation is going to drive several reductions. So the first thing that happens is that acetyl-CoA is attached by this enzyme onto oxaloacetate. This has 4 carbons. This has 2 carbons. Put it together, and we've got a 6-carbon molecule called citric acid. So the guy who discovered this is Hans Krebs. The cycle is called the Krebs cycle in his honor. It's also called the citric acid cycle because of this molecule that's at 12 o'clock on the Krebs cycle. But it's also called the tricarboxylic acid cycle. And again, if you remember your functional groups, they're not a big part of AP biology, but this is a carboxyl group. This is a carboxyl group, and this is a carboxyl group. So 3 carboxylic acids, the tricarboxylic acid cycle, and I think that shows up somewhere on this slide. Um, so now there's a bunch of enzymes, 2 through 8, and what they do is they'll systematically oxidize citric acid. And they're going to use the high energy electrons that I said high energy electrons, but this is a reduced molecule. It's got chemical energy. They're going to use this chemical energy to reduce NAD plus over here into NADH. We've seen that before. We saw that in the link reaction. And it's going to take this molecule, FAD, and reduce it into FADH2. Dead battery, charged battery. Dead battery, charged battery. And that happens 3 times in the case of NADH. Other enzymes that are shown over here power a substrate level phosphorylation of ADP and phosphate into ATP. So there we've got our energy molecule that can be used to do work. And that's an immediate outcome of the Krebs cycle. Now, if you're in a college course, there's an intermediate phosphorylation that involves GDP to GTP. But again, at the AP biology level, you don't need to know about that. What you need to know about is that for each acetyl-CoA that enters the cycle, excuse me, I'm getting over a little cold. Each acetyl-CoA over here that enters the cycle, you get 1 ATP. Here it is over here. You get 1 FADH2 over here, and you get 3 NADHs. You also wind up snipping off a carbon dioxide over here, and everybody just go like this. When you breathe out, you're breathing out carbon dioxide and other stuff, but 2/3 of the carbon dioxide that you generate through cellular respiration come about over here. The other third comes about through the link reaction. So if you've ever wondered where that CO2 that you breathe out comes from, and of course, it's not just you, it's anything that does cellular respiration. And all of those other things, all of those other organisms do it basically in the same way. And that's not a coincidence. That's because you are cousin to the plants that do cellular respiration and put out carbon dioxide. They also take in carbon dioxide through photosynthesis, but you are the relative of those plants. The mushrooms that grow from the soil, those are your relatives too. They do cellular respiration. They put out carbon dioxide. Well, you're their relative as well, even the bacteria that are in the soil. So life is a beautiful braided chain, and this is one piece of evidence for that. Let's see if there's anything else on the slide. Oxaloacetate is the starting and ending compound. I would put good money down that the things that I put in bold are the only things that in an AP biology class you would ever have to memorize. Excuse me. You'll have to memorize that acetyl-CoA brings chemical energy in. It's combined with oxaloacetate, which becomes citric acid. And then you'd have to know that NADH and ATP and FADH2 are the molecules that come out. So take a screenshot of this slide or watch this again on YouTube. And this is pretty much all you need to know about the Krebs cycle. So any other questions before I go on? Anything else about the Krebs cycle that anybody wants to know? Okay. I have a song about the Krebs cycle, which goes like this. Okay, enough of that, but I do want to tell you that this song is also available as a karaoke version. So imagine how much fun it would be if you were able to sing this through karaoke. Ah, there it is. So there's the karaoke version. And in the same way as you could sing glycolysis, and think about how you would just be the coolest kid in your neighborhood, wherever you live. If you live in Brazil, if you live in Des Moines, it really doesn't matter. And I just, of course, put a country in comparison with a small city in Iowa. Not a cool thing to do. But wherever you live, if you could sing these songs, you can impress your friends, you can impress your teacher. I don't know what else they'll think about you, but anyhow, it's a great way to learn, is really what it comes down to. So know that I have karaoke versions of all these songs, and go ahead and learn them in whatever way works for you. And also know that, I think I'm going to show you this, that I have interactive tutorials about this material on my website, learn-biology.com. Let's take a peek at that. And there we go. We're going to be able to get back. Fantastic. So this is the main landing page. If you go over here, you look at the courses, go to AP Biology version 2.0. That's the course that I've written for you. It's completely aligned with the College Board's course and exam description. And in the College Board's eyes, this is a textbook. You can use it. If you were a teacher, if I have any teachers who are watching, you can use Learn-Biology. You can put that down in your AP Bio audit, and the College Board will say, way to go. That's fantastic. You're using an approved resource. So you would go to the third unit, cellular energetics, and right over here, we've got an interactive tutorial about glycolysis. So I've got the song, and the whole thing is interactive. So you would log in and you would click Start Quiz. It's going to ask you who you are, and I didn't set the settings exactly right, but it doesn't matter. So here's like kind of a full fill in the blank thing. And like, even if you didn't know anything, you could reason it through. Glycolysis is that's got to be the first stage of cellular respiration. And if you got it wrong, it would tell you. It wouldn't let you put that. I'll do something wrong on purpose. Like that. See? It's red over there. It occurs in the cells. Oh, there's only one possible answer. It's the cytoplasm. Okay, so you're getting feedback the whole way through, and everything that we know about cognitive science in the brain says that this kind of interactive learning is 1,000 times more effective than passively reading a textbook. Songs are pretty good, I have to say. But you would go through, you would take this quiz, you'd get more feedback, you do a little bit more reading, and then you'd have a quiz like this. And I'll set the settings correctly. Which letter indicates the step where glucose 6-phosphate is rearranged and phosphorylated, becoming fructose 1,6-bisphosphate? That looks like it's letter D over there. I'm going to try that. Oh, I meant to put D. I put F, but look, I'm getting feedback. And that's the whole point. If you interact with the material and you get feedback from it, you learn about 1,000 times more. So if you're able to do so, you can get a free trial of my website, and then you can pay for a subscription. And if you do that, you will really, really, oh, thanks so much, Toxic. So you get a free trial on the website, and right now it is either $49.95 or $59.95 to use it for a full year. That, of course, takes you through the AP bio exam. Doesn't sound to me like that's a huge amount of money. So I would encourage you to just go and tell your parents right now, hey, there's an incredible resource out there. It's not very expensive, and it's going to help me get a 4 and a 5. And in fact, we offer a money-back guarantee on this. In other words, if you finish our course, and finishing our course is not easy. It's a lot of work. It would involve doing a lot of quizzes. But if you finish our course and you don't get a 4 or a 5, just send us a report showing us the work that you did, and we'll happily give you your money back, because this thing works. I used it with my own students. So Toxic, thank you very much for asking that question. Really appreciate it. So anyhow, you can learn about Krebs, you can learn about glycolysis. The whole thing is here. I've got tutorials about all these things. Next would be the link reaction, another interact tutorial with all the diagrams that I just showed you. And then finally, the Krebs cycle is right down here. And again, think about how much you'd have to learn or how much you'd have to know if you could do this. Like, this is becoming something else. This has 6 carbons. This has 6 carbons. Nothing could be happening other than putting like NADH over here and NAD plus over here. So anyhow, now you know we've got this website, and I totally encourage you to use it. If you wanted to use it, you would just go to, see if I can get this right. And yeah, there it is. So here are the prices. We have an app, $19.99. We have a website, just the website, $14.95. If you want both, it's $59.95. It's the greatest deal in education. So I hope you do it. Let's go back to our tutorial. Okay, so enough about that. Let's talk about the electron transport chain and oxidative phosphorylation. And we're right on time. I wanted this to go for an hour, so we're doing great. And I also want to encourage folks, any question about anything that's AP biology related, I'm happy to stop this and talk about that. My number one priority is that this is a live presentation. So feel free to ask away. And I'm really grateful to the folks who have put comments in the chat so far. So we're going to finish the process by talking about oxidative phosphorylation and the electron transport chain, which are two just unbelievably cool processes. One thing I feel really grateful for is that as a biology teacher, I get to teach the coolest stuff. So, explain how the mitochondrial electron transport chain generates ATP. We're going to walk through this over a series of slides. So let's get the geography set here. This is a very enlarged view of the mitochondria. And here's the outer membrane. Okay, outside of that would be the cytoplasm. Here's the intermembrane space. It's the space between the outer membrane and the inner membrane, hence intermembrane space. The inner membrane has all of these things drawn along it. Those represent, for the most part, membrane-embedded enzymes. These are proteins that are built into the mitochondrial inner membrane. And then there's the mitochondrial matrix. Again, that's like the cytoplasm of the mitochondria. So let's see how this thing works. Through glycolysis, through the link reaction, and through the Krebs cycle, we've been generating NADH and FADH2, which I've been encouraging you to think of as a charged battery, loaded with energy. Those are accumulating in the matrix on account of the link reaction of the Krebs cycle, and they wind up diffusing over to the inner membrane. And when they get there, they interact with enzymes that are embedded in the inner membrane. What they do is they oxidize NADH, rendering it into NAD+. They oxidize FADH2, rendering it into FAD. And I see a question. Is the reason for NAD plus being reduced into NADH so much because it is used for further processes in cellular respiration? If not, so I think that I just explained that. There's some very, very deep biochemistry here that you don't really need to know for AP biology. What you do need to know is that these molecules, NADH and FADH2, are mobile electron carriers. They're little batteries that can move around in mostly the mitochondrial matrix, but they can also make it from the cytoplasm into the mitochondrial matrix. And their function is to bring electron energy to this series of proteins, which is called the electron transport chain. So that's why we are in other phases of cellular respiration, creating NADH and FADH2, it's because that's charging up the batteries that are going to power this process that powers ATP synthesis over here. And if that wasn't a good explanation, then please ask me a follow-up question, because again, I live for this, love these questions, so keep them going. So here we're taking, again, our charged batteries are highly reduced electron carriers, and we're oxidizing them so that we can bring electrons. That's what these little E's are. They might be a little hard to see on your screen over there, but these are little E's. And those are going to flow along this yellow arrow, and that yellow arrow represents the electron transport chain. Now, electrons flow through the electron transport chain, which is a series of membrane-embedded proteins in the mitochondrial inner membrane. I have always encouraged my students to think about these proteins as constituting a little biological wire. A wire that can carry current. So these are the batteries. They're providing electrons. The electrons flow along this wire. Now, in the devices that you use in your life, your phone, a radio, maybe if you still use that, anything that you use batteries for, right? Well, the batteries supply the energy, they supply the electrons, and they power a device. Well, the device that they power is shown at number 3 over here. Those are protein pumps. I said that wrong. They're proteins that are proton pumps, and their name says exactly what they do. They take protons. This is a proton over here. Proton is, the nucleus of a hydrogen atom. It's a positively charged particle, and there's physics there that we don't even need to begin to get into. But it takes protons that are in the matrix and pumps them into the intermembrane space. That is active transport. We're pumping from low concentration to high concentration. And I see a question from Toxic. How much ATP is generated at the end of the UTC? We're going to look at that and add it up all together. But I think that if you add up the ATP that's generated from glycolysis, Krebs, and the electron transport chain, we get about 32. Because this is a biological reaction and not strictly a chemical reaction, the number actually varies. So it depends on optimal conditions. It depends on which transport protein the NADH that's generated in glycolysis uses. So if you leave this lesson knowing about 32, that's about as much as you need to know, again, for an AP Bio class. But I think that if you dug into Campbell biology or something like that, you wouldn't get a much better answer. So let's see how that ATP is generated. And that's perfect. So 26 to 28 from the electron transport and 30 to 32 for all of cellular respiration. So fantastic. Great job. So where were we? So we've got a wire that's powering a device. The device is these proton pumps, and they're pumping protons from the matrix to the intermembrane space. That's an energy requiring process. It's not powered by ATP, as most energy requiring processes are. It's powered by electron flow. So this little electrical current that's powered by the food that you eat is generating this proton gradient over here. And the way that I always encourage people to think about this is a biological fuel cell. It takes a fuel and it uses it to generate an electrical current that powers a device. Just so that you know, the other important analogy is that, photosynthesis is like a solar panel. So this is like a fuel cell. And what we've done is we've created an electrochemical gradient. Now, those electrons are only able to flow because of oxygen. So oxygen is the final electron acceptor in the electron transport chain. I'm circling it over here. And what oxygen does is it also soaks up protons out of the matrix gloms those protons onto oxygen, creating a molecule of water. When you breathe out, you're breathing out water vapor. Now, some of that is just, the water that's in your body, but other water is what's called metabolic water, and it's created through this process. There are rats that live in the desert, kangaroo rats, and I believe that most of their water actually comes from metabolic water, since it's hard for them to find water in their desert environment. So here's the role that oxygen plays over here. So we've now powered, I think I sort of jumped around accidentally. Yeah. O2 acts as a final electron acceptor, pulling electrons down the electron transport chain, increasing the proton gradient between the intermembrane space and the matrix. So protons are disappearing over here, whereas they're accumulating over here. Now, these are all positively charged particles. They don't like being with one another. Positive charges repel one another. So they're all desperately looking for a way to get out of the intermembrane space. They can only do it through one channel, and that's called ATP synthase. And its name tells you what it does. It synthesizes ATP. The kinetic energy of diffusing protons through this channel is used to power the synthesis of ADP and P into ATP. It's called oxidative phosphorylation because oxygen is required for the electron transport chain, and the electron transport chain is required for this gradient, and this gradient is required for this facilitated diffusion that enables the synthesis of ATP from ADP and phosphate. That is the astoundingly cool process by which cells generate ATP from food. Now, you can sing about this through one of my best songs, which is the Electron Transport Chain song. And I'm just going to play a little bit of it. And I'm going to try and jump to the chorus because the chorus really sort of gets it all. Let's see. Here we go. I don't know if I have that memorized, but I know that students have memorized it, and they've told me that their ability to memorize, let's see if I've got it, the mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons kinetic energy to make ATP from ADP and P. That got them a 5 on the ATP exam, on the AP Bio exam. This song actually goes into more detail than you need to know. There is, let me see if I can get there. I got a karaoke version of that. If you can sing this, then you're just, the cat's meow. That would be really cool if you could pull that off. And I didn't mean to do that. What I really meant to do is this. I mean, here you are. I want to encourage you to sign up for a free trial. And I just want to say, if you sign up for a free trial, the worst thing that can happen to you is you get on my email list and I send you a helpful email with fun things about things that I observe in biology, or I send you little explanations of biological features and stuff like that. I was on a bike ride and I saw this peacock in the Oakland Hills. I live in California. I'm going to write about that pretty soon. Oh, I think I might have already done it. So just anybody out there already on my list, I'm just wondering, just give me an I am if you're on the list. I'd love to see. I'm not seeing anything come in, but it takes a little while. There's a little bit of latency. So really the one thing I want to encourage you to do is I'm putting out videos all the time because I want to get you ready for the AP Bio exam. My mission is really to um, conquer biology confusion and to help people deeply learn biology. And on the way, I really want people to get a 5 on the AP Bio exam. It's why I created learn-biology.com. I created it for my own students, and now I'm sharing it with the world. I want to encourage you to do it. So go ahead, sign up for a free trial. Don't delay. It's what it's what my purpose in life is all about, helping people learn biology. And really, I hope that you are on a similar kind of mission, which is to learn biology, because it's the greatest, most fantastic, most mind expanding of substances. So we're about to land the plane. I planned on this going from 7 to 8, and I don't know what time it is where any of you are, but anyhow, are there any questions? Anybody have any questions about anything that you want to ask me about? Yes. Um, and thank you very much. So there is another process. I wasn't going to go into it today. I did cover it on a recent movie that I just released. It's a review of all of Unit 3, and I'll just jam forward there. Um, it covers aerobic respiration and fermentation. So if you want to see me explain those processes, just go to that movie, just go to Science Music Videos and just look at recent videos. And I have one about all of Unit 3. And actually, pretty soon, I'm going to be breaking those videos apart into smaller videos. So you can watch that separately, or you can watch this here. I want to thank you so much for spending your time. I know that this universe is full of wonderful things to do, and there's also homework that you've got to get done, and who knows what other things you're involved in. But you chose to spend your time hanging out with me and letting me do the thing that I love most, which is teaching biology. I really hope that I see you up on learn-biology.com. I want to deeply, deeply encourage you to sign up for a free trial. If you're motivated, if you're a warrior who's getting ready to get a 5 on the AP Bio exam, then this is your path. It's the most efficient way to do it. I'll be here again in a couple of days doing another live. I'll probably do them on Wednesday, not Tuesday, but I got to figure out my schedule. But I'm going to try and do this on a regular basis. So again, I want to thank you so much for taking your time to be with me tonight. It's really been a joy to get to teach some biology to folks around the world. I want to really thank those folks who chimed in with comments, because, man, it makes me feel great to know that people are actually watching. And thank you so much. I'm going to see you again soon. Everybody, just know that I'm very, very grateful. And I'll see you again at the next stream. Check out the channel. I'll put another notice about when it happens. If you're on my mailing list, I'll email you about when the next live show is going to be. Everybody be well. Thank you so much. And take it easy. Have a good night. Bye, everybody.